have Dr. Rob Ellis, who's going to talk to us a little bit about some of the ideas of how you create the right environment in aquaculture for culturing a range of species. So Rob is a lecturer at the University of Exeter in Biosciences, and he did his PhD in 2013 in the University of Plymouth, which is just down the road from where I was brought up as a young man. So we've been kind of talking about some places like Rugby Salt, Holman, my old stumping ground, which is rather nice. But apart from that, Rob will give you a great talk here about how basically aquaculture can be designed, the animals within aquaculture can be designed, and some of the stresses.
Thank you very much, Chris, and thank you for welcoming me. So as Chris said, I'm a lecturer at the University of Exeter. I'm actually also a research fellow, a NERC, Industrial Innovation Research Fellow. So NERC in the UK is the equivalent of the NSF. And I'm an Industrial Innovation Fellow because of the recent problems with Brexit and the UK trying to find themselves economically and forge a strategy. And there's a pot of money to develop a product, or to do a research product that develops a product for industry that can improve the UK economy. And I've kind of pinned myself on aquaculture. And genetics, and I'll introduce you to that a little bit later in this talk, but as a background, I'm actually an ecological physiologist. And did my PhD, as Chris said, at Plymouth University in the ecological background, looking at physiology of mussels, and how mussels adapt to climate change, ocean acidification, and warming. And over the kind of past 5 to 10 years, I've shifted much more into a commercial focus, how we can understand about meeting kind of aquatic protein production demands. And the talk I'm going to kind of walk you through is the way I think about how I approach research and how we might do this from two different viewpoints. But first I'm going to set the scene a little bit. So what drives me in my research? I suppose addressing this major challenge, how do we meet the global food demand that a growing global population that's projected to be 10 billion by 2050 is going to increase? And it's been projected that we need to increase food production by around 70% compared to today's production levels to feed the world in 50 years. What makes this challenge even more intractable is we have to meet this challenge in the face of a global climate changing at a rate unprecedented for millennia. So it's not just that we need to produce more food, we need to produce more food in a world that makes it difficult to produce more food. This is a really interesting conundrum, and I think scientists globally are tackling this. Now, I might be kind of biased, but I think there's no environment in which this is more evident than in the marine realm. Having to produce more food to feed the growing population, as well as maintain biodiversity and the challenges that climate change produce. Now, as a species, we're hunter-gatherers traditionally. We've traditionally caught all of our food from the wild in order to feed ourselves. Now, for agriculture and land-based agriculture particularly, this shifted around 10,000 years ago. So that's when we started to domesticate animals on land to start to mass produce. We've been very successful at that and actually have a very established agricultural industry. However, this isn't the case in the sea. And up until sort of the late 80s, early 90s, we caught almost exclusively all of our fish for human consumption from wild fisheries. Now, over the last sort of 3 decades or so, there's been a major shift. This has come around from 2 different areas. Firstly, the flatlining wild capture fisheries, and the fact that most of the stocks globally are now overexploited or at a maximum sustainable yield. Secondly, aquaculture represents the fastest growing food production sector globally, and it has done for 3 decades. So in fact, we've passed a really critical threshold in 2014. This was the point where aquaculture actually produced more food for human consumption than wild capture fisheries, and it was the first time it did so. More importantly, with wild capture fisheries flatlining as I projected, and it's projected they can continue to do so, aquaculture offers the only viable way to sustainably increase production to meet the growing demand for seafood globally. So it's a really important sector with which to increase aquatic food production. But the capacity of this sector to do that and meet the raise the challenge remains kind of really uncertain. And that's because of a number of challenges that face the sector. I've already introduced climate change being one. The other is disease challenges. Can we do this? And the sustainability of this sector. How can we maximise the sustainability of aquaculture to ensure we meet the aquatic protein demand? Now, to put this in context, it's often been pitched this as a future challenge. We need to increase food demand by 2050. But actually, a recent paper by the FAO showed that within the next 5 to 10 years globally, we face a 28 million tonne deficit in seafood production and demand. And this is because of a growing middle-income sector. They're becoming richer and therefore eating more seafood, as well as paired with a growing global population. This means we're going to need more seafood to feed a growing population. What's really quite harrowing in kind of this challenge is the fact that that report didn't account for the effects of climate change. So that's almost the best case scenario. So in fact, we're facing at least a 28 million tonne deficit in seafood within the next 5 to 10 years. Now, we'll all be very well aware of the benefits for human kind of health and nutrition that seafood brings, as well as poverty alleviation, and the key role it plays in terms of the sustainability of communities and their viability and an income. Therefore, any deficit in seafood production can have a real dramatic impact on the global population in terms of an ability to maintain healthy populations and reduce adverse health outcomes. And this is particularly evident in developing nations and small island states. There's a huge global challenge that needs to be addressed. And as a physiologist, I'm really keen to kind of see how we can use different skill sets to address this and how we can approach this. So with my background from climate change, I'm trying to, my research really takes two different approaches. Is how can we address the seafood deficit? The first is how can we optimize the environment to suit the species? So if we think about aquatic food production, just like on land, it's quite an intense environment. It's intensive production. How can we optimize those environments that are very different from wild environments to maximize productivity and reduce adverse health outcomes and make the production as sustainable as possible? Obviously, though, this is only possible in an environment which you can manipulate and control to some degree. Now, this isn't always possible in many production sectors, and I'll introduce that in a minute. So another area that we're starting to look at is how you can optimize the species to suit the environment. So this is more analogous to some of the research Chris is doing, is how you can use selective breeding approaches to really kind of manipulate the stocks of the animals you're growing to ensure that the species you're producing and the strains of those species are specially adapted and able to cope with the environments that we're producing them in now, as well as the environments that we project to be producing them in within 50 to 100 years, so looking at future environments. And can we make sure that the stocks that we're growing are able to produce food that we need now, but also in the environment that we're predicting to occur in the future. Firstly, though, I'm going to introduce this first topic, focusing on optimizing the environment and the kind of the concept that we're thinking about that, how we can really think about optimizing this environment. Now, this research came about from a project I was involved in about 10 years ago, which was a UK ocean acidification research project. And I was coming from a climate change background, just out of my PhD, looking at how ocean acidification and global warming is going to impact mussel physiology. And we kind of expanded that slightly and thought, how can we project what aquaculture in the UK might look like in 50 years? And there was a growing body of research from the climate change field at that time to show you need to understand the baseline variability in different environments to then project what's going to happen in the future. So with that in mind, we set up a program working with a number of partners across the UK. So what we did here was we worked with fin, fish and shellfish aquaculture producers, both on land and in open ocean conditions. And we first taught them about ocean acidification and the problems that they might be facing due to climate change. And then we taught them about the benefit of understanding carbon chemistry. We then taught them how to sample the water within their facility and enable them to do so over an annual cycle to give us a baseline of the current production settings in each of these different environments and then how we might project the future conditions. So as I said, it was really a baseline setting experiment for climate change studies, or that's what we pitched it as. But actually the reality turned out to be far different. Now, unfortunately, I haven't got time to go through all of these examples one by one, but I'm going to highlight two of them specifically. The first of which is the Menai mussel strait. So this is one of the key mussel growing regions in the UK. It's in kind of the Isle of Anglesey in North Wales. It's a small island separated from the mainland Wales by a tidal strait. It's only about a mile wide at its widest, and it gets much narrower in certain places. And it's a very highly dynamic environment. So there's water that runs through this strait. It's changing direction each tidal cycle. It's fully marine, so there's no dramatic freshwater input in this system. And it's really productive in terms of mussel production. So what we asked the farmers to do was to sample on a relatively coarse scale. We didn't have the sensors to do this, so this was all manual sampling. It was just getting a very crude, basic understanding of the variability over kind of seasonal cycles within this, in this habitat. And as you can see here on the temperature, you've got a nice seasonal temperature cycle. It's quite cold in this habitat. But the salinity is very stable, so there's no freshwater input, there's no real fluctuation in salinity. The alkalinity is nice and stable, which is reflective of that. But But PCO2, for example, is a little bit higher than perhaps might be predicted from this environment, certainly from what was known before. And in fact, in this environment, PCO2 exceeds 600 microatmospheres around 40% of the time. Now, this just goes to show these environments are probably more extreme than would be predicted from open ocean and climate change scenarios, but then that's nothing new. But really interestingly here, aragonite saturation is below 1.5 around 85% of the time. Now, to bring aragonite saturation, as I'm sure you're all aware with George and Chris's work, is a metric that shows the vulnerability of a shellfish production area to changing climatic conditions, with aragonite saturation being really important for organisms to make their shells, particularly for bivalves. So here we're showing that it's below 1.5, 85% of the time. So this would suggest that actually this population is particularly vulnerable to any shift in climate conditions. And therefore, any change in carbon chemistry could be particularly detrimental for production in this region. Moving on to the next example, we're now going to a lobster hatchery. Now this is a really nice example of a community-led initiative. So the Padstow Lobster Hatchery is a charity based in Padstow in the very south of Cornwall. And the idea is here they optimise the fishery for the community. So what happens is fishermen collect buried females, they'll bring them back to the facility where they're maintained in tanks until they release the larvae. The larvae are then released and the females then fin clipped and put back out into the wild to then go through a number of cycles. And this notching then means that they can't be harvested for subsequent cycles. The larvae are then grown out until the larvae settle and are grown through a few stages, with the idea that you're providing an environment in which you're protecting them from predation, so you can maximise survival and productivity of those larvae for the subsequent lobster generations. They're then returned to the seabed via a really weird reverse Hoover mechanism, so they pump them back onto the seabed with the idea to maintain lobster populations for future generations to fish and to make this a really sustainable fishery. However, again, looking at the environment in which they're being grown, as you can see, PCO2 here would certainly not be kind of benign, certainly. PCO2 averaged over 700 microatmospheres, around 15% of the time, exceeded end-of-century projections. So we're growing up lobsters here in an environment that we're calling optimal for their growth, but actually with respect to PCO2, we're moving away from what you'd certainly call optimal in a climate change setting. Indeed, aragonite saturation state in this time was below 1.5, 70% of the time, and indeed it's below 1, 36% of the time. So just like the mussels, these lobsters are calcifying and making a hard shell, and in fact we're bringing them into an environment where that might be very much more energetically expensive than would be expected in the wild. So therefore the whole premise of providing an environment that optimises productivity of these larvae could be a miss, and actually we might be growing them up in an environment that while there's no predators, actually could be detrimental to their physiological state. And again, these are conditions that the lobster hatchery were completely unaware of, and they're occurring kind of asymptomatically. But it might be that there's a vulnerability to the system that could be switched if this condition is deteriorating any further. With this mentality of an increasing kind of intensity leading to a deviation from natural conditions, we decided to add a further site or a further partner. Now this is a land-based facility, again on the Isle of Anglesey in North Wales. It's a recirculating aquaculture system, so it's entirely land-based. And there's a partner called Anglesey Aquaculture. To give you an idea of the scale of this place, the picture here is of their main facility, and one of these circular tanks, in scale, these are 16 metres across and 5 metres deep. They have 8 of these in sequence, and at any one time during maximum production, each tank can house 200,000 sea bass that they're growing up to market size. So it's a very intensive production setting. And at the time of sampling, this was the largest RES in Europe. So what is an RES, a recirculating aquaculture system? Well, the premise here is you remove growing animals from their natural environment onto a very controlled land-based facility. The one in Anglesey that we worked with had 10,000 tonnes of seawater in the facility, and it's constantly being recirculated, but only 1 to 2% every day is renewed. That's still a large volume of water that's being replaced into the environment, but in terms of the facility itself, that's a very small percentage of water that's being changed. Now the idea of RAS as an aquaculture setup is that it reduces the water use and the environmental footprint, but it increases the biosecurity and the stability of the chemistry within any one system. So it's supposed to be environmentally friendly, and it's really argued as the forefront of aquaculture. where aquaculture should go to in terms of making it as sustainable as possible and have the least impact. Now, as with all kind of intensive production systems, there's a number of challenges here. So to walk you through the system, the tanks up here, the three, is where they keep the animals very intensively in their setup. Now, obviously, our animals are very productive. They're growing, so they're breeding. And they're withdrawing down oxygen and producing CO2 and producing toxic ammonia. Now, aquaculture systems on land are very well adapted at coping with many of these challenges. For example, there's very vigorous aeration, often bubbling in pure oxygen. So oxygen saturation often maintained at 100% saturation or even above this. They've got very established water treatment, biological filtration that can remove toxic ammonia and other nitrogenous wastes. CO2, however, is an issue that they're currently not understanding and they haven't gotten to grips with. And in fact, some of the things that they're actually doing management-wise could be making this worse. So in fact, bubbling in pure oxygen, for example, to reach your 100% saturation threshold, when you're bubbling pure oxygen requires 5 times less gas than if you're using air. Therefore, you're off-gassing 5 times less CO2. So therefore, this leads to a build-up of CO2 in the system that they're not overcoming. And yet, because they aren't measuring CO2 directly, they're not aware of this challenge. So with adding in this partner, we looked at the facility to try and understand the water chemistry and the challenges that they're going on. Now, aquaculture is very well aware of the issues of pH for growing animals and the challenges of low pH impacting productivity. And they measure pH readily. And if you were to focus on pH solely, if you look at the inlet seawater compared to the seawater in the tanks, this would seem exactly as a system running in optimum. They're maintaining pH exactly where you would expect. But if we focus on CO2, actually CO2 is around 3 times end-of-century projection. So this is certainly not alleviating any of the carbon chemistry issues. And actually the root cause of the issue, of any productivity issue in this system, is due to this build-up of CO2. Now, how are they achieving that? Well, they're doing so by adding in alkali. So they're buffering the pH because they know they need to maintain a steady pH, and they do so by adding in alkali. And this is causing additional problems. Now, in fact, they're adding so much alkali that it's around 8,400 micromoles per kilogram. And as far as we're aware, this is the highest system, the highest level of alkalinity that we've noticed, and we've certainly not seen any natural analog. So they're growing fish in an environment that's completely atypical. And as far as we're aware, there's no data showing what impact that high level of alkalinity has on the fish that are growing for things like nephrocalcinosis and other kind of adverse health outcomes that have been noted in many finfish production settings. The other thing is this is a real problem in terms of human health. This is how they're doing it. They're using 25% caustic soda, sodium hydroxide, and they're pumping in 3 tons of this into their facility every day. So this is a human health issue because it's very caustic, so you're having to handle this stuff and pump it in. And bear in mind, I come back to this later, but this isn't one of our most high-tech facilities in Europe. This is even more of an issue in other regions we've worked in. Sorry. But what they're doing is they're delivering this directly into their filtration system. Adding in that much caustic soda causes precipitation of calcium carbonate. So the white at the bottom here isn't foam, as you might expect, but it's actually a little mini mountain of calcium carbonate in their pumping station. So every now and then they turn off the adding of the sodium hydroxide, somebody jumps in and they have to chisel it off to clean it so their pumps keep working. So that's a kind of more work that they have to do. But in addition, if you look at things like calcium, seawater has a calcium level around 10 millimolar, but they were precipitating out calcium here, so it's now down at 6 millimolar. Now, anecdotally, there's research has shown that a level in seawater fish of 4 millimolar can be lethal. So they're approaching lethal levels of calcium for seawater fish in this facility completely unawares because nobody's ever measured calcium in their facility. So again, there's very little research to show what kind of detrimental impact this might be having on the production efficiency and other areas. And therefore, understanding this and then being able to undertake the experimentation to optimize this is really important for this system. Just to note that actually they added sodium hydroxide, you can use other alkalis. So we suggested they switch from sodium hydroxide to sodium bicarbonate at one point, which would stop the precipitation of calcium. They found a much bigger change. Now we suggested sodium carbonate as well. They were less willing to take that on as a cost thing. But it's talking about how you can demonstrate the benefits of management effects that can really help the industry in terms of efficiency and overcoming some of these adverse effects. So to kind of finalize this part of the the story, we then decided to extend this to a further part. So I went out to Thailand a couple of years ago to work with some partners to look at how, if this is the case in the UK, what about other extreme environments where we're producing animals? So this was in collaboration with some universities, the University of Mahidol, for example, as well as Centex Shrimp, and also CP. So CP are the largest producer of Thai shrimp globally. And they provided us access into a number of their facilities. And what we wanted to do here was sample the variability and the carbonic chemistry across different pond systems. So we wanted to look at spatial variability and temporal variability in different shrimp systems. And to do this, I sampled in 2 separate regions. Now, firstly, focusing on spatial distribution, this was something I undertook myself. So I sampled 10 ponds across 4 different farms. At each time of the day, it was always at noon, so the temporal variability shouldn't be that great. And then the difference here will be different in terms of different chemistries or production levels or within these different farms, so different developmental stages, for example. And what you can immediately see here, across these 10 different ponds from this single region, there's quite significant variability in terms of carbon chemistry. So PCO2 here, for example, I've highlighted, it averages just under 2,000 microatmospheres. However, this can vary from just below equilibrium with the atmosphere, so around 330, up to just shy of 7,000 microatmospheres. So this is in the same region, same kind of pond, just different levels of production intensity. So it shows that there's a real variability in terms of different shrimp ponds that we need to capture if we can understand the stresses that these animals are growing under and then mitigate against it. And as you can see, again, the alkalinity is quite variable here. And that's because, again, these Thai shrimp farmers on earthen ponds wearing flip-flops and shorts are throwing in the same caustic soda, or often caustic soda flakes in this case, to mitigate pH in their system. So they're doing exactly the same as the high-tech system in Wales, but with much less health and safety. So being able to understand this could be a real benefit for human health. We also then looked at temporal variability. So to do this, I asked a farmer to sample for me from dawn to dusk, because I didn't have access to a farm myself at this time, in 3 ponds on 1 farm. So this is a single farm, so the same source of water, 3 separate ponds, and they sampled from dawn to dusk, sampling every hour over 4 days. So essentially on the first day you're sampling 4 hours separate gaps between each sampling point, and then the next day you'd start an hour later and carry on. So you capture the dawn to dusk over the 4 days. And just to highlight on the graph here, the dark cycles, or the grey bars, represent the night, so that's when the farmer wouldn't sample. But that's not a problem, and certainly the data we were able to gather has given us a really interesting picture. But what you can see here immediately is if you look at the green, blue and red dots, they are each colour represents a single pond. So you can show that each pond is very different in terms of the variability, in terms of carbonic chemistry, which was shown from the previous table. So you can each pond has a different dynamic that we need to understand. But the other thing is how variable it is over a single 12-hour cycle. If we look at the green pond, for example, panel 1, this single pond had a PCO2 rate of 2,200 microatmospheres in a single 12-hour cycle. So here we're talking about climate change with animals growing, and then we're going to have 1,000 microatmospheres by the end of the century, or as upwelling might be occurring with 1,600 microatmospheres. But in this system, you've got a shrimp that's having a 2,200 microatmosphere fluctuation every 12 hours. So this is caused by a build-up of CO2 during the night. So the algae and the shrimp in the pond are respiring at night, producing CO2, and then in the day, as the sunlight kicks in, the algae become photosynthetic and draw all that CO2 back down again. So it's constantly cycling through this, and the intensity of it really depends on the productivity within that pond. But really interestingly, I actually presented this data at a conference in Bali about 3 weeks after I sampled the data, and the farmer who had taken it for me was at the conference and came up to me and said, okay, do I know what colour pond represented what pond system? I was like, no, I did it completely blind, I just know green's green, red's red. Why? And she said, because actually immediately after you sampled, we had a die-off from white feces disease. Now, white feces disease is a bacterial disease, I believe, that causes the epithelium of the shrimp's gut to completely slough away. And then they release white feces, and symptomatically about 12 hours later the entire pond dies. And they had that break out in one of their ponds. It's not causative, but that happened to be the green pond. So the one with the highest PCO2 variability had a mass die-off immediately after the sampling. Now, as a physiologist, it's really intriguing, and it actually starts to beg questions. Should we be sampling carbonic chemistry more and other pond variables to be highlighting kind of adverse health outcomes and kind of the system of the pond? If they'd have been measuring PCO2 and knew that high CO2 variability was indicative of an outbreak, could they have harvested just before the outbreak occurred and then have not lost the investment that farmer had made in that single pond? Which for a small-scale farmer with only 4 ponds, who puts a cycle through every 90 days, is huge. That's a quarter of their productivity. So there's little things that we can do as physiologists that might help give an understanding to the industry to become more productive and tackle some of these challenges. So that's the data that we've been able to collect and understand carbonic chemistry variability. But what about the wider ecosystem and aquaculture generally? What's known? Well, actually very little. So doing a literature search, we looked at the available data in terms of carbonic chemistry data available for aquaculture productivity and aquaculture systems. So salmon aquaculture is one of the largest sectors globally. It's a huge business, billions of dollars. If we look at the salmon smolt industry in Norway, a paper by Chris Noble in 2012 showed 40% of salmon smolt hatcheries averaged a PCO2 of just shy of 5,500 microatmospheres. So that's the 40% average to that PCO2. Actually, some of them went up way above this to around 17,000 microatmospheres. So this is the average level. So here we go to one of the most sophisticated and established aquaculture industries globally with all the technology has a very large CO2 issue that until very recently they were unaware of. So one of the most rudimental and basic Bangladeshi shrimp ponds. Again, there's a couple of papers that have shown Bangladeshi shrimp ponds again average around 17,000 microatmospheres of PCO2. And again, there's only a handful of papers that have shown this because there's just not the data out there. There's not enough data out on the variables within ponds to understand the extreme conditions that they're operating in, which then gives us an understanding of the kind of the parameters the organisms are coping with physiologically, with which we need to understand to help optimize. And for certain examples, like sea pens, there's a complete lack of data. So there's no data on carbonic chemistry within sea pens. Certainly that's not widely available. I'm sure if you went to speak to the live producers like Marine Harvest, they have their own data, but they keep it in their back pocket and they don't share it. So this is problematic in itself. In a sense, as a kind of a physiologist, it's very difficult to compare these fields and understand the conditions organisms are coping with. So why might this be a problem, and how can there be a contrasting perception of CO2 in climate change fields? We're kind of ingrained to think anything above 1,000 is quite extreme. But obviously aquaculture here is occurring, and to the large extent is being very productive and successful in a much more extreme setting. So just to kind of put that back into context, here's a figure from a paper I published a couple of years ago. End of century in climate change is around 1,000 microatmospheres in the open ocean. We're around 400 present day. So how is there such a disjunct between what we're showing in aquaculture, the conditions, and climate change? Well, the first issue with being able to compare between these two fields is the confusion of units. So climate change and physiologists typically use partial pressure, which is usually microatmospheres. And this is because a partial pressure is what drives the diffusion of CO2 across a gradient, in this case the animal's gills. And that's the CO2, therefore, that the animal is experiencing. In aquaculture, they typically use a mass concentration, milligrams per litre. Now, there's a real issue with this in terms of being able to compare between these two systems. And that's because it doesn't take into account the solubility of CO2 in water. To demonstrate this, again, this is from the same paper, we showed that each point on all of these 5 coloured lines is 1 milligram per litre. So it's exactly the same CO2 concentration in milligrams per litre. However, if you go from freshwater cold, that can be as low as atmospheric 400 microatmospheres. If you follow the green line with the dotted at 10 degrees, that's 400. However, if you go into a highly saline environment that's quite warm, so a shrimp production system in Saudi Arabia, for example, this same level of CO2 can be at end of century predicted levels in the partial pressure. So this, for an aquaculture scenario, these are the two climate change point of view, the level of CO2 that the animal is actually experiencing and is driving its physiological processes are the two ends of the spectrum with respect to climate change understanding currently. So this really brings in a challenge in terms of comparing aquaculture and climate change and understanding what's currently known between these two fields. And the first thing we implore trying to bring these two fields together is to present both both parameters. So presented both in microatmospheres and a milligram per litre. So you can compare between these systems. The second is in terms of why there might be a disjunct in comparison between these two fields, is the control levels. As climate change scientists, current atmospheric is control, it's 400 microatmospheres. Of all the aquaculture studies we looked at, the lowest control level was 1,000, typically closer to 3,000 microatmospheres was their control level. So therefore, all of their impacts that they're comparing to are to what we broadly deem end of century or very extreme. And therefore, there's a possibility here, certainly, that within climate change, they've gone beyond the threshold in terms of measuring outputs that we might be picking up in climate change studies that they're not noticing because they've gone beyond that threshold without what we would call a true control. Now, I'm not pointing the stick at aquaculture saying this is bad, because actually, in these scenarios, it's often impossible to achieve a true control level. You can't get rid of the CO2 as these systems are so extreme. But it really begs a question about how we can compare the two fields. But really, what is apparent between these two fields is there is a striking difference in the perceived impact or importance of CO2 for productivity and for adverse health outcomes that really need to be addressed. And this is really intriguing as a physiologist, is how this can be the case. Two very different fields, two very different thought processes on the same stressor, which ultimately is impacting the organisms in the same way. How can that be the case? And what learning can we do by bringing these two fields together? And again, why might this be the case? Well, aquaculture has a benign environment. So there's no predators, for example, certainly in a land-based facility. Often their animals are fed out in the beaten or as much as they want to do. And to a large extent, they try and minimize disease as much as possible. So whilst CO2 and carbon chemistry and other abiotic parameters might be very extreme, there's other factors that might be influencing the functioning and the physiology and biology of the animals. It means that stress can be coped with. There might be some impact with the different endpoints measured. As physiologists, we look at subtleties and nuances. Some of the major research coming out of climate change research, certainly from the finfish side point of view, is looking at kind of cognitive neurobehavioral stuff, looking at sensing, olfaction, auditory processing. Those aren't important for aquaculture necessarily. If a fish can hear a predator or smell a predator, it doesn't matter. The food's thrown in front of their face. They don't have to try and find it, and they don't have to run away from the predator. So do we care that they might be going swimming around, not sensing the food because there's enough there for them to get? So there's different endpoint measures that are really important in these two fields, and actually having an understanding of that is critical. And trying to get some alignment between what the two fields are looking for with their respective research. And something I spoke to with Chris a little bit earlier is, actually, is there a domestication for selective CO2 tolerance? It's been occurring inadvertently. So coming back to the salmon example, for example, you've got 2 or 3 decades of selection for the best producers in a salmon production environment. So they've been producing optimum growth in these environments. Now actually they've been unaware of the CO2 issue. But is it that the CO2 issue has always been there and they've been selecting for a CO2 tolerance that they've been unaware of? So these animals are now tolerant to CO2, whereas they wouldn't be as a natural population. There's that possibility of domestication to CO2 inadvertently. But as climate change scientists, we could really use that information. We could learn from that by then going back and looking at that pedigree and understanding what selection's gone on, and therefore using really clever approaches to say, we've got a 20 or 30 year adaptation experiment where you can compare salmonella tolerance to CO2 based on that exposure. But really, does it matter? And this is kind of the big take-home message. I think as researchers, we should all think about this. Does it matter? Obviously, does it matter that climate change shows subtle impacts or quite extreme impacts of CO2 at often lower levels, yet aquaculture is productive under much higher levels? And the bottom line is productivity. Is a business productive? Now, over the last 5 to 10 years, I've been really enjoying kind of the back and forth between like commercial production and academia, and that dialogue, and really making sure that as a scientist I'm addressing questions that the industry needs to address to make themselves be profitable and viable and to help address the food challenge. And again, the productivity and the profitability is the real key issue. And getting that framework of thinking as a farmer who's trying to produce food, we're talking about climate change at the end of the century, but really we should be framing these questions within sort of 5 to 10 years. can a farmer remain productive within the next 5 to 10 years, because that's kind of the timescale over which they're thinking of their business and the next steps and mitigation strategies. And if you go to a farmer and say in 50 years or 100 years climate change might stop you getting spat, you won't have a great deal of traction if the projected forecast in the next 5 to 10 years is they're going to still remain profitable. But if you can show that actually with very subtle changes in the next 5 to 10 years you can dramatically increase productivity, or if you don't do this, you're going to have an issue, then you have a little bit more traction and you can work with them. And now coming back to kind of climate change literature and where this can really be beneficial, it's talking about adverse health outcomes. So talking about the impacts of high CO2 in these environments, I'm going to focus on 3 very quickly. So this is disease, product quality, and production efficiency. So these are 3 metrics that the aquaculture industry are very well aware of and very interested in and want to overcome. So with respect to disease is a $6 billion kind of impact on agriculture globally every year. So every year, $6 billion is lost due to disease. So therefore, from a kind of a simple economics point of view, if you can overcome the issue of disease, that's analogous to increasing production by around $6 billion of new production. So we don't actually, if we can tackle the disease issue, you don't need to produce ponds big enough to produce 6 billion tons of productivity. You just stop the disease, and that's the same overall impact on productivity. So what can we learn about disease from CO2, or what is already learned? So some of the research that I've done during my PhD and research I've undertaken at the same time has shown that actually CO2 can have a really negative impact on disease within an aquaculture setting. Firstly, this is research by a PhD student, Elizabeth Dorfmeyer, from I think it was San Francisco State back in 2012. And it just so happened she did the exact same PhD as me in America when I was in the UK, where she took the pathogen point of view and I took the host point of view. We used the same pathogen, this is Vibrio tubiaceae, and she looked at the impact of CO2 on the pathogen growth. And the take-home message here was under CO2s that are certainly shown in aquaculture conditions from what we've looked at in the literature and from what we've done from our monitoring. The Vibrio tubiaceae is more prevalent in terms of there's higher colony-forming units within these conditions, and also more virulent. So under higher CO2 conditions within an aquaculture context, we've got a disease here that's certainly more abundant and more virulent. So therefore this could cause a disease issue. My PhD research showed that actually with mussels, high CO2 causes a reduction in immune defense. Now the population we used is very resilient, so you have to use quite significant CO2 levels to drop the pH to have an immune effect. However, these are certainly conditions we've noted within other aquaculture settings again. So they might not be relevant in a climate change point of view, but certainly in an aquaculture setting point of view, these conditions can be achieved, and therefore, with elevated CO2, you have a host that's immunocompromised, and you can have a disease agent that's increased in virulence. So that's kind of like the perfect storm. But that's only really half the picture, because actually a disease-host kind of dynamic is really about the host-pathogen interaction, what happens when these 2 things come together. So we did a research in Sweden to look at this. So again, the same host, same pathogen, mussels and Vibrio tubiaceae. But we didn't go anywhere near as extreme with the CO2. In this case, we only went to pH 7.7. Now, to walk you through this, in this experiment, we didn't find any measurable impact on the host immune defense. So if you were to stop there, you'd say, okay, there's no impact on elevated CO2 on the host itself. And we didn't find any real noticeable impact on the Vibrio. So again, you just said there's no impact on the pathogenicity. So therefore, your logical conclusion would be there's no impact on host disease interactions. But as you can see from the graph here, when you infected mussels grown at high CO2 with bacteria grown at high CO2, there was a far higher count on the ability of those bacteria to colonize those mussels under the future conditions compared to any other condition. So here, even in the absence of any notable impact on the host or the pathogen in isolation, elevated CO2 is impacting host-pathogen interactions. And therefore, again, considering CO2 is really important in terms of disease implication for aquaculture, and something that might be being massively overlooked presently in the environments where CO2 can be very variable. Shifting very quickly to production efficiency. So this, I'm going to present some research here by a group in Norway, Basco-Water. And as far as we can see, this is some of the best research that's been undertaken in terms of looking at production efficiency in a commercial scale where the CO2 is really properly parameterized. So this is a huge group out of the FEMA. And they've run an experiment where they looked at PCO2 impacts. Again, you'll note the control level is 2,000 microatmospheres, So yeah, 2,500, so it doesn't come anywhere near to current atmospheric. But fascinatingly, they found a linear growth decline under elevated PCO2 of around 24%. So if we kind of take aquaculture industry and growth efficiency trials and those kind of things, if you're looking at the feed industry within aquaculture, if you can have a 1% impact on growth efficiency for a feed company, that is huge news for the salmon industry. And there's billions of dollars being plowed into that to make it as productive and efficient as possible. So showing that there's a 24% growth decline with no other adverse impacts noted under high CO2 is very big news here. This is a really important story. And again, where CO2 is not being widely monitored, it is largely going undetected because those fish that are coming out that are 24% smaller have largely no other noticeable impacts of this elevated CO2. So it is very conceivable that aquaculture can be proceeding, growing things at high CO2 and not noticing it because the fish are relatively healthy, just they're growing smaller. With that said, this study also looked at gene expression. And what they found was under the two different CO2 conditions, the 2,500 microatmosphere and the 20,000 microatmosphere, around 70 genes are differentially regulated in response to high CO2. Critically, of those 70, 40% are related to immune function. Now, they didn't do any disease trial or disease susceptibility in this group, but again, it begs the question, is CO2 kind of critical for disease susceptibility? Understanding that is really important for mitigating against disease in aquaculture. Finally, I'm going to talk about product quality, and this is a really interesting study by some colleagues of mine, again, the guys out of Sweden, and it's looking at the northern shrimp. And what they looked at was the impact of CO2 on product quality. How good is it? Good is it true? It's a really clever experiment they designed. Essentially, they did an ocean acidification experiment, grew up two cohorts of shrimp, one under endocentric conditions, one under current conditions. They then brought in a professional chef, a celebrity chef, to cook them up, and they had a blind taste test panel to taste the shrimp, which was a great idea. And consistently, these shrimp were graded lower quality in terms of taste, texture, and appearance, consistently by all of the taste test panel. So this makes a really interesting question. The biggest take-home message that always gets traction in the industry is critically 29 out of 30 of those taste testers would pay more for a better quality shrimp. So when you go up to a farmer and say, look, CO2 might be negatively impacting your ability to sell a shrimp at a higher price because they're a lower quality, that really rings true and it gets their attention. And from a consumer's point of view, it really asks the question, are we already eating substandard seafood because it's being grown under intense conditions? And might this be a way of kind of leveraging research and a kind of almost a certification to improve the quality of seafood by doing some kind of organic mark, saying actually you can produce seafood under better welfare, lower CO2 conditions, and you can actually market it as being better quality because the research out there supports this in terms of CO2 productivity and actually the quality of the shrimp. So that's kind of a whistle-stop tour of my research in terms of aquaculture environments. I realise I'm approaching the end of my talk, time allocation. So I'm going to very briefly whiz you through my current fellowship, which is taking the adverse approach. So how you can, in environments that you can't manipulate the environment itself, like you can on land, how can you optimise productivity? And this is focusing on the species. And to do this, I'm focusing on mussel hybridization. So my fellowship is a 3.5-year fellowship funded by NERC, and it's focusing on the three Mytilus species we get in Europe, so Mytilus edulis, Mytilus procellus, and Mytilus galliprovincialis. They have a really complex speciation web. So they occur in Europe, California, hence why I'm here, and in Chile. And the species coexist and readily hybridize. So it offers a really interesting model to investigate the role of hybridization and how that controls environmental tolerance and species adaptation to different environmental conditions. And as I said, they occur in different regions globally, so the ability to then test this same question in Europe and European populations, as well as in Californian populations and Chilean populations, offers a really interesting opportunity to look at evolutionary ecology. These regions all adapted at different evolutionary scales. Europe's hybridization matrix has occurred over several thousand years. California's over several hundred, and Chile's over several decades due to human introduction. So can we look at the ability of the species to hybridize and then traits to be conserved across regions by looking at this model? And just to highlight the fact, I'm here working with UC Davis with Antogen. I'm also collaborating with Recirculating in Chile. So the approach I'll be taking is I've sampled 24 populations across these different global regions, and we're going to be sequencing them to look for genetic markers of local adaptation, or SNPs. I'm then going to develop a genetic tool, a SNP array, which will then be able to type mussels globally, and this is working with a partner, Ross Houston at Roslin. And at the very beginning, when I talked about the fact that I'm on the Industrial Innovation Fellowship, and I needed to produce a product that could benefit the industry, and this is my product, and this is why I got the funding. And I'm not a geneticist at all, so this is really outside my area of expertise. But what I then had to do is to look at physiology. So as a physiologist, I want to look at performance in mussels. How you can differentiate high and low performance, how consistent is performance? Do you have generalists versus specialists? And is that consistent through an individual's life cycle, or is it consistent through an annual cycle? Look at how that individual copes with stress, different stresses, can they cope with a single stress very well, or can they cope with a broad range of stresses? And then compare that back to the genetics, which can now enable us to start asking some really interesting questions in terms of local adaptation and evolution to different stresses. But then could be the starting point for our aquaculture industry to start looking at selective breeding and trait selection in mussels if we're starting to think about this selective breeding to optimize shellfish for the future. And as part of that, I've got 2 PhDs running, one on selective breeding and barriers to that. This is the PhD student who's taking a slightly different approach. So she's looking at selective breeding from the outside. So assuming everything's perfect with the genetics, of course it's not. We don't know everything, but assuming we did, what do we do between broodstock conditioning and grow out that might inadvertently negatively or positively impact selective breeding success in terms of things like grading or feed concentration or the environment in which we're growing our larvae, which will better able to make selective breeding occur, be more successful or not by accidentally knocking out part of your genetic variation by inadvertently grading at a certain point through the production cycle and then having an impact that you're unaware of. So she's doing this working with collaborators across the UK, and she's working across a number of different biological species to answer questions within that framework. The other one, the project I was just going to highlight is kind of my last point, is another PhD starting this September. We're looking at shrimp production in this case. And again, it's going back to the RAS. So there's a massive movement globally to put shrimp production on land on the outskirts of cities. Now, so the CP has 90% of Thai shrimp production. They've actually just invested on a facility just outside Florida. They're going to be building an RAS to produce shrimp, so they can produce it and then ship it to the city. And if that model works, they've got an idea to do that all across the United States, where homegrown food is a real key kind of leverage tool at the minute for the Trump administration. And they're looking to expand that. So we're looking at how you can optimize the environment in terms of to produce shrimp in an RAS, where that physiological data is currently unknown and the environmental conditions are currently unknown. So is producing a shrimp you take from the pond going to be suited to RAS, or can we produce or optimize the environment and the shrimp species to suit this new environment being established for shrimp productivity? So in summary, to wrap up my talk, from what we've shown from our kind of research over the past 5 or 10 years, aquaculture conditions, certainly with respect to CO2 and carbon chemistry, are often extreme, certainly taken from a natural environmental perspective. There's often no natural analogues and there's high variability. But most importantly, they're very poorly understood, particularly with CO2, due to the fact that CO2 is very difficult to measure accurately and reliably on a ground commercial scale at an affordable price. So this makes it quite tricky. But physiology might offer us the tools to help improve water quality, to select the boundings and the optima in which to make aquaculture as productive as possible and minimise adverse health outcomes. And really bringing these two communities together can be really fruitful to help tackle this food security problem. And then with a nod to my fellowship, certainly hybridization may offer a mechanism to understand kind of environmental tolerance and offer that kind of local adaptation. And understanding it can be really beneficial for shellfish breeding in the future. And it certainly builds on some of the really good quality research that's come out of Chris's lab and others in the area. And with that, I'd like to say thank you very much for listening, and I'm happy to answer any questions that you might have.
Got time for questions?
So
So can I invite you to tell us again what the tool is that you got your fellowship for and what it will do? I'm sorry, I wish you had all the talk to talk about the genetics. I'm a geneticist.
I know. So essentially, it's a SNP array. So we're currently doing a sequencing approach where we've taken 24 populations of mytilus from across the globe, but they've been selected specifically on the environments from which they've come. So instead of going out and picking mytilus HMSC pure, Gallo pure, and Trosylus pure populations, we've chosen populations from temperature gradients across each of those three regions. We've got the Baltic climes, we've got a salinity climb which goes from HMSC to Trosylus. We've got upwelling regions in each of the three continental areas. We've got a population that's tolerant to harmful algal blooms. We've got a population that's tolerant to heavy metals. So those are all currently being sequenced at the minute, which is why I've got 3 months out from the lab at home to be able to come out and have my visit to the US. But then essentially what we're going to be doing is selecting the genetic markers that differentiate between those populations, so the SNPs. We're then going to create a SNP panel. So it's essentially that plate has 50,000 markers that you can still. So instead of doing a whole genome sequencing approach, which costs a lot of money, each muscle can be typed on that plate for around £15, I believe. And you'll get the kind of what allele it has at each of those markers instantaneously, essentially. So it's a cheaper way to type muscles within a population. So essentially, the very minimal that will do is it will offer a farmer a way to understand the genetic composition of their population. It will tell them the speciation that they've got within that population, and it will offer them a mechanism by which for food traceability, which in an increasingly consumer-aware market is really important. So for them to be able to test their muscles and then go to a supermarket somewhere that's claiming to be selling their muscles and show, okay, that is my muscle like stock or it's not, it's really important. So that's the minimum the tool will do. Beyond that, it will offer the application. There'll be many SNPs on there that aren't kind of associated with the speciation that can be used to interrogate local adaptation. So we can use it for genome-wide association studies, looking at these kind of physiological tests that I'm doing in the lab and looking at actually what's the profile of different performance within the lab. If you have a higher level performer physiologically, genetically, what does that look like? And does it marry on to what we might predict from local adaptation, the markers that we're finding locally? So it's starting to, it's kind of like a two-pronged approach to say what's driving this tolerance of certain populations. Is it genetic? Is it plasticity? Could it be other things, epigenetics? We'll all undoubtedly be having roles, but if there's any benefit to selective breeding within muscles, this will certainly start to pull out some of those beneficial traits or groups of linked markers that might be helpful for
that. You suggested that some of the ways that aquaculturists are operating are driving ECO2 levels higher. What sort of options would they have to reverse that trend and actually get back to lower value?
It's always the one they don't want to hear. It's lower density. So don't have as many animals in your system. Don't push it to the nth degree, but obviously that's not the aim of the industry. So it's a really fine balance. And I think we need to develop technology to be able to first measure and understand CO2 in the system so they're aware of the conditions that they've currently got. But then it's about an ability to strip CO2 back out, and particularly using land-based RAS is a really key example here. We're working with a couple of companies in the UK, firstly to see if we can get cheap, reliable CO2 sensors that can go into aquaculture settings that are cheap enough but give you a kind of a reliable enough measure of CO2 within PCO2 that you can understand it and see how variable it is over time. At present, some of the cheapest sensors you can buy are around $17,000 for a single sensor, which is fantastic from a climate change point of view, but for an aquaculture point of view, that's not even on the question. So looking if you can get cheap, reliable technology there would be fantastic. The second is then optimizing the CO2 stripping technology. At present, that's all built on kind of counterflow current, off-gassing with air, and kind of Venturi systems to remove CO2 from the water. But that's very inefficient. So research we're currently doing at the minute is trying to understand the efficiency of that and the different options on the table and how you can improve that efficiency of existing technology. We're working with engineers to try and develop some more technology that might be better, have a higher efficiency. But then it's thinking about new approaches about how can you strip CO2 out of aquaculture. And it kind of along the lines of some of the stuff you're doing with adults is how can you integrate multiple strains? Can you do aquaculture with algae and other species simultaneously? And actually, if you can light the algae 100% of the time, then you can have a system that's actually actively removing CO2. And might that take the edge off this CO2 buildup that might just help with some of these problems? So it's going to be a multi-pronged attack for sure, but always being mindful in the back. Kind of as a scientist, certainly, it's easy to say, okay, removing the CO2 would be relatively easy if you increase your flow rate and reduce density, but obviously that's not working within a business mentality. So it's trying to think around the challenge, but in a way that you can still achieve their optimal goal of being as productive as possible.
One thing I thought was really interesting is both your PhD work and this student in California showing this interaction between disease and CO2 levels. But in both your studies, it looked like your high PCO2 level was like around 2,000 atmospheres, whereas, in aquaculture, looking at a range that's like up in the 10,000, sometimes higher levels. And has anyone else looked at kind of the impact of these really high aquaculture levels on disease?
So my the PhD student was up around 2,000 microatmospheres, but had disease work in San Francisco State up with the Vibrio. My stuff actually went up to about 18,000 microatmospheres with the mussels. We looked, it wasn't pitched with aquaculture in mind at all. It was more actually a carbon capture sequester and CCS leakage scenario stuff that we were doing at Plymouth at the time and looking about what would be the impact of a catastrophic leak. But it just so happened we hit on a level that's being shown in aquaculture. And the mussel population I use, that's actually really resilient. They're very good at coping with low pH conditions. They're in an estuarine environment where PCO2 fluctuates quite readily. So we had to push them to that extreme condition to get any kind of noticeable response. And actually, going to the climate change literature during my PhD, we got a lot of kickback from people saying, actually, these conditions never occur. Why are you going so extreme? This is kind of pie in the sky, mechanistic, it's not very important. And now kind of reel on 10 years down the line, I'm like, actually, well, no, here it is. We found the conditions where it's relevant, and actually it just happens to be for a commercial species. So it's actually coming back to be our benefit. But mussels, it is that level, but there's not a lot out there. Very few people are doing research on scenarios where the CO2 is taken that high, just because up until recently, aquaculture wasn't aware it had this CO2 issue, and climate change, unless you fitted with the kind of guidelines from EPOCA where they said you should only go to 1,000 microatmospheres, and if you go above and beyond, that's not relevant. There's a big gap in our understanding that needs to be filled in terms of all these different ranges in terms of both magnitude and variability that we haven't got the answer to yet. But hopefully some of the stuff we're showing suggests it's a pertinent area to study.
So going back to the green shrimp pond, how do you account for the rapid increase and then subsequent decrease in CO2 level?
That's all to do with, so at night, the pond's got a very heavy algae load. So at night, the algae and the shrimp are respiring, so the CO2's skyrocketing. But then as soon as the sun comes up, the algae switch to photosynthesis, and they've got much more biomass in the pond. So despite the fact the shrimp are still respiring, they're just drawing all that CO2 back down. So during daylight hours, the CO2 comes down to just below atmospheric, and then at night, everything's respiring and it comes straight back up again. So that's where the cycling is, and the differences between different ponds is all to do with different biomass levels of algae and shrimp, and there's lots of different ways of doing this production and how green they make their ponds. So, and obviously they're not permanently lit. If you could light them 24/7, this wouldn't be an issue, but you can't do that for the energy impact.
If there are no more questions, thank you very much.